inches long. So I went ahead and put a little bit of alcohol in the area, and I palpated that. Just palpate again really quick. Okay. All right, and there we go. We have a sterile sample. All right, so we have our urine sample here. Really quickly, we're just going to go ahead and assess it visually. The color, it's a little bit more yellow, slightly more concentrated, I would assume. It can be anywhere from a clear, where it's fairly dilute, to a really dark. It can be even darker than this. The volume-wise, we had, the bladder wasn't too big, wasn't too small. It seemed like a normal volume. We only drew up 3 mLs of urine. The clarity, it looks pretty clear. I don't really see it too cloudy or anything. I can see straight through it. I don't see a whole lot wrong there. Odor-wise, I haven't really smelled it yet, so whenever I put it on for the specific gravity, I'll just kind of see if I smell a bad odor. Whenever animals have a bad odor, sometimes that also you can tell if they have a urinary tract infection or something going on there. So I'm going to go ahead and get a specific gravity on it really quick. I'm just going to make certain not to touch my syringe to anything. Just place 1 drop on my refractometer. Alright, and it's greater than 40, so I'm going to go ahead and just rinse this off really quick. Alright, so I have that cleaned off now. And let me write down my specific gravity. I'll just put greater than 1.040. Alright, so our next step is putting the urine onto our sample so that it can run through our urinary analysis reader. So I'm just going to place a drop on each of these little test strips. It's going to run the pH, the glucose, it'll see if there's any blood in there. And once again, just making certain not to touch my syringe onto the sample. Alright, so I'm going to place this into our reader. And I already have the animal's information entered in, so I'm going to hit start. That's going to go ahead and process those, and then we'll get the answers, and I'll go ahead and relay those to you here in a second. Alright, so we have our urine results back now. The pH was 6.5. The protein registered as +1. Glucose was negative. The bilirubin was +1. The ketones were negative. Blood, it did not register any blood either. That was negative. Specific gravity, once again, was greater than 1.040. So what we're going to go ahead and do now, our sample finished spinning, so we're going to go ahead and get that smeared out and stained. Alright, so we're going to go ahead and spin this urine down so we can go ahead and look at the sediment underneath our microscope. I'm going to go ahead and open the top of the tube here, and then squirt some of my sample into the tube. I don't want to place all of it in there. I want to keep some in case we need to send that off for a culture. So I'm going to place that there. I have my other test tube here, about the same amount pretty much, so I'm just going to go ahead and place this into our centrifuge, and I'm going to go ahead and place that on for 5 minutes. Alright, so we're now going to go ahead and get our urine smear made. So what I'm going to go ahead and do is just drain out the excess urine so that just this material here on the bottom is remaining. I'm just going to gently kind of shake that back and forth, mixing up what was in there. I think that looks pretty good. I'm just going to grab this. So what I'm going to go ahead and do is just put 1 small drop here on my slide. And then I'm going to go ahead and smear that out. Alright, so I'm going to go ahead and let this dry, and then we'll stain it. Alright, we're now going to go ahead and stain our urine smear. I set it out to let it air dry, and so I'm just going to go ahead and dip this into our Dip Quick stain. We have the 3 steps here, so I'll let it sit for about 30 seconds in the blue, which is the first one, then I switch it to the second and then the third. Alright, so after we get done staining, we're going to let this dry, and then I'll go ahead and take a look at it underneath the microscope to see if I see any bacteria, blood cells, casts, crystals, etc. We'll see what all is on here. Alright, so I'm going to go ahead and switch over to the purple now. Put it on here. Alright, so I'm going to go ahead and just gently rinse this off, and then I'm going to let it sit out to dry. Alright, so I now have my urine slide to go ahead and take a look at underneath the microscope. It's all dry, so I'm going to start off by looking at it under 10. Alright, I'm going to switch it up to 40. Let me try that again. That wasn't really working. There we go. Okay, now let me switch it over to 40. Alright, I have it zoned in 40. I'm going to go ahead and put my oil on here, and I'm going to go up to 100. Okay. Alright, so I'm just going to kind of go over this, see if I see any casts or crystals, see if I see any bacteria, blood. Alright, so the urine sediment that we spun down and stained turned out to be normal. Didn't find anything. There weren't any casts, no crystals, no blood, no bacteria, didn't see much white cells, no red blood cells. So that's not surprising. We had just ran it to go along with the CBC chemistry panel. So the dog wasn't having any urinary symptoms or anything, but we still like to spin ours down and double check those. So now I'm going to go ahead and go over some commonly seen crystals, casts, etc. That you would see in the slides. Really quickly, first of all, I want to go ahead and go over the red blood cells. Red blood cells you commonly find in urinary tract infections. So I will zoom into that picture a little bit. Alright, in addition to that, you also have white cells, which are commonly found as well. And here is a good example of a white cell. Alright, so it's commonly, in addition to bacteria, you'll find red blood cells and white blood cells. Here are some glial cells right here that can be found in urine, and I think I actually have a couple of other pictures as well, some additional pictures of what it might look like, just for an example. And then, in addition, also bacteria is something that is commonly found. You'll find cocci and rods in urine. Here we go. Here is a good example of what cocci and rods would look like on a urine sediment. In urinary tract infections, you'd want to go ahead and send that off for a culture so that you can make certain that you're using the appropriate antibiotics to go ahead and treat those. So, in addition to that, we also have casts. Let me pull up some of my pictures of casts. Casts I don't see that frequently. here's kind of a good example there. Castor scene, you know, that has more to do with the function of the kidneys, and they come from the tubules from the kidneys, so with those loops. So I don't see those very frequently. One thing that I really like are crystals. We see those fairly commonly, and triple phosphate, otherwise known as struvite, is very common. I'll kind of focus up into that. Here's a couple of these ones. So we have triple phosphate, which is also known as struvite, or magnesium ammonium phosphate. You have calcium oxalate dihydrate, calcium oxalate monohydrate, ammonium urate. Those are commonly found in Dalmatians and are very, very painful crystals to have. There's a couple of other ones on here. Calcium carbonate is on here. You don't see that one very often. Bilirubin, tyrosine, cystine. So those are all some of your common crystals that you may find. Depending on the pH of the urine will have a lot to do on what kind of crystals you would find. Crystals can be treated through diet, just by changing the food that they would consume. So you commonly see those with cats that come in that are blocked. One thing that you would want to make certain too, whenever you have animals that come in with urinary tract infections or crystals, you would want to take an abdominal radiograph to see if they have a bladder stone in there as well. But red blood cells, white blood cells, bacteria with the cocci or the rods, crystals, cast, you can see protein in the urine. Those are all commonly found things in your urine sediment that you would observe on a slide.